Let's now move on over to hear from Dr. Jianming Hu, who's a professor in the Department of Microbiology and Immunology at Pennsylvania State University College of Medicine, where we'll be going and giving us a overview of hepatitis B virus replication in natural history. Jianming?
Okay. Thank you, John. First, I would like to thank John and the organizing committee for inviting me to this interesting seminar webinar. So I was asked to give a brief introduction to the replication cycle of hepatitis B virus and the natural history of hepatitis B. Next, please. So hepatitis B virus, of course, has been known for a long time. It has, interestingly, multiple types of particles. For virus, of course, it has an intact virion. That's what's depicted on the top right. It has an envelope, and then with viral proteins, 3 viral envelope proteins. Inside the purple layer is the viral capsid, composed of 1 viral protein, the core protein. Inside is the viral DNA with the viral reverse transcriptase or polymerase attached to it. And then also depicted is a protein kinase or PK that's incorporated from the host cell into the virion. Okay. In addition, there are also spheres and filaments sort of depicted at the bottom of the left. So spheres, just about 20 nanometers long, as opposed to the virion, which is about 42 nanometers in diameter. And the filaments is the same diameter as the spheres, except as it says, it's a filament. So both spheres and filaments actually are not intact viruses. They only have envelope proteins. They don't have capsids, and they don't have DNA. So what's also depicted on the right side is a more recently discovered and characterized type of particle. We call it empty virus or empty virion. So it has an envelope on the outside, and it has the purple layer, which is the capsid, but it has no DNA and no polymerase. Interestingly, it also has the protein kinase, also incorporated from the host cell. Next, please. So this slide depicts sort of a simple diagram of the viral genome. Again, in the innermost circle is the partially double-stranded DNA. We call it relaxed circular DNA. So on the outside, what's called a minor strand, which is complete. In fact, it has a little repeat. On the inner side is so-called the plus strand, which is incomplete, as depicted by the dashed line. And so within this very short genome, it's only about 3.2 kb. The virus encodes 4 open reading frames of 4 genes, and from those 4 genes produce 7 viral proteins. Most of which I already kind of talked about. For example, the 3 envelope proteins, S, M, and L. The polymerase, which is the longest viral gene, encodes the viral reverse transcriptase, or RT. And then capsid protein I mentioned already, but then on the same open reading frame of the capsid, there's a N-terminal extension. So if the N-terminal extension plus the capsid region that consists of the pre-core protein. And then finally, we have the X protein. Okay, so all these proteins sort of are depicted on the outside of the partially double-stranded DNA. And then on the very outside of this diagram, those wavy lines, those are the viral mRNAs. Okay, so the longest one is called 3.5 kb RNA. It's also called the core RNA because it's used to translate to produce the viral capsid protein as well as the polymerase protein. In addition, it's also called the pregenomic RNA or called the pgRNA because it's the precursor to produce the viral relaxed circular DNA by reverse transcription. So in addition to that 3.5 kb RNA, there are also the 2.4 and 2.1 kb RNA. Those two are translated to make the viral L protein and M and S protein, respectively. And then for production of the viral X protein, there's a separate xRNA, the 0.7 kb RNA, So what's also depicted here is all the viral RNAs have the same 3' end that terminate at the same polymerization site. So indeed, the viral genome is very highly compact. So every open reading frame overlaps with every other open reading frame. And all 3 viral envelope proteins, for example, are completely embedded within the polymerase gene. And the regulatory sequences are depicted, for example, the enhancer 1, enhancer 2, the basal core promoter or BCP promoter, PRES1 promoter, PRES2, S promoter, and the X promoter. All those are actually overlap with the coding sequences. So it's a very compact genome. Next, please. So this slide depicted the sort of the viral life cycle. So we know a lot about this, but we also don't know quite a bit about this also. Okay, so the virus, again, is an enveloped virus. So it enters the cells through binding to the viral receptor called NTCP. But then afterwards, the entry process is actually not very understood. That's the step 2, depicted with a question mark there. But ultimately, the relaxed circular DNA from the virus is delivered to the host cell nucleus, which is then actually repaired by host DNA repair system to form the so-called covalently closed circular DNA that we already heard about, which is really the basis for viral persistence. And we'll hear more about this later at this webinar. So from cccDNA, the virus then takes advantage of the host RNA polymerase II and transcription factors to produce all those viral RNAs that I just talked about. Those viral RNAs then are exported outside the nucleus to produce the viral proteins. And as I mentioned, the reverse transcriptase protein, the capsid or C protein, the X protein. And the RT and capsid protein then comes together with the perigenomic RNA that forms the so-called immature nucleic capsids. So that's step 6. So in there, within the immature nucleic capsids, the pgRNA is then reverse transcribed to make a single-strand DNA. Then from the single-strand DNA, making a relaxed circular double-strand DNA. So that's step 7 there. Okay. So once the relaxed circular DNA progeny is made inside the cytoplasm, it can actually recycle back to the nucleus. So that's step 8. So that's the so-called intracellular recycling or re-amplification. So what that does is that in the infected cell, a single nucleus not only just contain the cccDNA come from the infecting virion, but it can also come from the progeny rcDNA de novo made in the cytoplasm. So you can have more than one cccDNA in a infected cell nucleus. Okay. Well, once the viral envelope proteins, again, there's 3 of them, LMS, reaches certain levels, then the mature capsid containing the rcDNA, those like the broken hexagon, are directed to outside the cells, forming virions. So that's step 9 there. So that finished the whole cycle. Okay. So what's also interesting recently people found is there's a low level of pgRNA before it's actually reverse transcribed. So those immature capsids can also be enveloped and secreted as RNA-like virions. So that's step 9 depicted there, okay, with a question mark, okay. And more interestingly, also like we found that the capsid can form actually an empty capsid inside cells without packaging the RNA or the reverse transcriptase. So those capsids are obviously dead end, but they can also be enveloped by the LMS protein and secreted as empty virions. So again, the empty virions have an envelope, has capsid, but has no genome. Okay. So in addition, on the right-hand side, there's a depicted spheres and filaments, which I already kind of talked about. So what's important to notice is that spheres and filaments or surface proteins can be produced from cccDNA, but in addition, can also be produced from the integrated DNA. Okay. So the integrated DNA comes from double-strand linear viral DNA, which for simplicity I did not depict there, But in the cell, there's always a small minority of double-stranded linear DNA, which can be integrated in the host cell genome. So that's step 3A there. Okay? And the integrated DNA can be produced, can be transcribed to make the surface RNA. So that's step 4A. So again, the integrant-derived RNA can be made to produce L and MS protein. So what that says is then the spheres and filaments in the blood or outside cells is not only reflecting cccDNA transcription, but also integrated DNA transcription. So that obviously has important implications for a diagnostics or biomarker. Okay. Next, please. So in addition, actually, could you go back? Sorry. So in step 9C on the very right-hand side, there's the pre-core protein I mentioned, right? So pre-core protein is just slightly longer than the core protein. But the pre-core protein is not usually part of the capsid. Instead, it's secreted outside the cells as a soluble dimeric protein, originally thought to be just this one protein called E-antigen after proteolytic processing. But more recently, we and others have identified actually a related protein called pre-C, which is very much related to the E-antigen with a slightly different processing. Next, please. So this slide just shows a little bit more detail of the 3 viral envelope proteins. Again, these are produced from the 2.4 and 2.1 kb RNA. So all 3 viral envelope proteins have the same S domain. In fact, the small S protein has only the S domain. But then the M protein, in addition, has the pre-S2 domain. And the L protein then, in addition to that, has the pre-S1 domain. Okay? So, and each one of these have a non-glycosylated form as well as a glycosylated form. So the glycosylated form is called GP, or glycoprotein. Next, please. So here, I just want to bring out some recent structural studies that a collaborator of ours and us published recently through cryo-EM methods. So it's known for a long time, HPV produces a huge amount of spheres and filaments. In fact, those are the basis of first generation HPV vaccine. But we actually did not have a high resolution structure until very recently. So just a couple of years ago, we managed to publish the first high resolution structure of the HBs antigen, the spheres, that's depicted on the right-hand side. It has a quite interesting structure. So one of the most striking things is, in contrast to what's depicted in the textbooks, these spheres, filaments, and in fact virions, they don't have a typical lipid bilayer. So in fact, HPV is not a classical enveloped virus at all. Okay? So this obviously has implications for virus entry and capsid release from virions, as depicted on the left side. If there is no virion envelope, there would be no viral cell membrane fusion, in contrast to what almost all the reviews or textbooks would depict. So what this says is the viral entry process is going to be quite different from what everybody envisions or depicted so far, based on the structural information we had recently. Next, please. So here, just a little more detail of the viral capsid and pre-core gene. Again, so the capsid is very small, just 21 kD depicted on the top there. It has an NTD, a linker, and a CDD. Okay, and again, the capsid is the major component of the viral capsid, which is also the major component of the complete virion or empty virion depicted on the right-hand side. Okay, so the core protein or P21 is produced from pgRNA, but then the pre-core protein is actually produced from a slightly different RNA called the pre-core mRNA, which is almost the same as the pgRNA, except it's a little bit longer at the 5' end. Because a little bit longer, then it has an additional upstream AUG, which allows the translation of a pre-core protein. It's about 39 minutes longer, the precursor. initially than the core protein. So that's the P25 protein depicted at the bottom. Okay. So from P25, then the signal peptide is removed. So now you have P22. Then from P22, the C terminus is removed, the red part. And now you have the so-called E antigen or P17. So that's the classical E antigen people have been detecting in the blood for decades. But then in addition to that E antigen P17, at the very bottom, there's also protein originally called the P22CR. Recently, we renamed it as a pre-C. It's very similar to the E antigen, except the signal peptide at the terminus is still not removed. So it's very interesting how that protein can be released from inside the cells. Okay. So again, most of the pre-core proteins are not part of the capsid. Instead, they are secreted as dimeric proteins, E antigen and the pre-C protein, as depicted on the right-hand side. Next, please. So here, just a little more structural information. So the capsid structure has been solved quite a long time ago, multiple times indeed. So the capsid basic structure building block is a dimeric. So it's a dimer of a P21. And then 120 dimers then forms a T=4 capsid. So what's depicted here is actually a C-terminally truncated capsid protein, which only has the NTD and the linker region expressed in E. coli and form the T=4 capsid. So in the CDD, which is missing from this crystal structure, it has the phosphorylation sites as depicted with those numberings. Unfortunately, it got shifted. The multiple phosphorylation sites, those high nitrated serine and threonine residues in both HBV and DHBV. And the CDD is only heavily phosphorylated and dynamically phosphorylated, meaning it's phosphorylated and also dephosphorylated. And it's involved in many functions of the capsid protein. Okay? For example, RNA packaging, DNA synthesis, nuclear localization, and there's probably also involved in regulating assembly, although it's not absolutely required. Okay. Next, please.
Jianming Hu, sorry to interrupt, but you have about 3 more minutes in fairness to the other
Sorry,
speakers.
okay.
So okay,
Yeah,
sorry,
I'll go much faster. Okay, so I just want
about 3 minutes. Thanks.
Sure. So the linker originally was thought to be just a spacer. Now actually we have evidence to show it's involved in multiple steps of viral replication. Next, please. Next, please. So here, I just want to say there's a little bit more complications than what I just been describing to you. Although the capsid is usually thought to be composed of a single protein, but recent evidence suggests that pre-core protein, including that P22 and P25, can be a minor component of the capsid protein. And in addition, another truncated capsid protein called HBC182, derived from a spliced RNA, can also be part of the capsid and then actually be important for viral infection. So it just means that the information we have so far maybe not really quite complete as we thought. Okay, next, please. So here I just want to say one word about the X protein. So X protein is involved in regulating viral transcription. The most recent or the accepted version of this is the X protein degrader host factor called SMC5 and 6. So SMC5 and 6 then cannot suppress cccDNA transcription. Okay, next, please. So in terms of cccDNA, I'm sure you'll hear more about this. I just want to go this over quickly. So the relaxed circular DNA is a broken DNA, then it has to be repaired to form cccDNA. Because of time, I'm not going to talk too much because you'll hear more later. Next, please. So this again, I think Dr. Guo, we're going to talk about this more. I'm going to skip. Next, please. So here, I just want to mention a few words about the virus assembly process. So I mentioned the pgRNA is the template for producing the capsid protein or C protein and RT protein. It's so the capsid protein, RT protein can then bind to a RNA signal on the pgRNA called epsilon. So RT binding to epsilon is the first step, and then the capsid binding to the RT and epsilon complex is the second step. So it's a two-step assembly process. Next, please. And it turned out this RNA package signal called epsilon is also the origin of viral reverse transcription, because the viral DNA synthesis actually starts at the 5' end at this epsilon signal. Next, please. And it's through a very unusual mechanism called protein priming, because the primer is actually the reverse transcriptase itself. And we think there are actually still many different ways to tackle this particular step, which remain to be exploited. For example, we've showed you can have a sort of a unusual nucleoside analog like clavidin can inhibit this protein priming step through a very unusual mechanism, non-competitive, and the nucleotide is not incorporated. And I think this is a very interesting mechanism that remains to be exploited for antiviral therapy. Next, please. Okay. So in terms of the capsid, I think we're going to hear more about this. So some of the evidence from our lab suggests the capsid protein is involved in cccDNA regulation. Okay, can increase intracellular amplification, for example, but then block entry. And then, of course, host factors also can regulate entry as well as intracellular amplification. And some of those host factors we think actually determines species specificity. So it has implications for animal model development. And obviously, this has been targeted as the antiviral targets. I think Dr. Zarkin is going to talk about this more. So go ahead, next, please. So anyway, so I think, again, we're not going to talk about the CAM because we're going to hear this again. Next, please.
So if you could summarize, please.
Sure.
We need to move on, please. Go ahead.
Right. So I think, go ahead. I mean, I just want to go to the next slide, please. So this is just a couple of structural slides showing the new information that we have this pocket factor on the viral capsid and how the DNA was organized, that may help us to understand how the rcDNA escapes the capsid to form cccDNA. Next, please. In terms of natural history, I think this is basically says it's a long process that provides opportunities for antiviral therapy, of course, for vaccine to prevent infection, and then we can prevent all the consequences. Next, please. Next, please. Yeah. So in terms of the natural history, I think the most recent identification is we sort of moved over from the previous classification of the 4 different stages, and now we have so-called E antigen positive infection, E antigen positive hepatitis, E antigen negative infection, and E antigen negative hepatitis with different virological and immunological characteristics. I think this new classification better reflects the immunopathogenesis. Next, please.
Yeah, thank you very much.